Hello, today's lecture is going to focus on organelles and their functions, and we're going to start off with some AP released questions to review, and then we're going to get into the actual content of the lecture. So the first question, at the end of each of these, I'd like you to pause it, try to figure out the answer on your own, and then I'm going to reveal the answer and the explanation in the following slide. So in this first one here, all of the following cell components are found in prokaryotic cells except DNA, ribosomes, enzymes, or the nuclear envelope. Well, the answer is D, nuclear envelope. Remember that there are 4 components found in all cells, whether they are prokaryotes or eukaryotes. They need some type of genetic information, some type of chromosome. They need ribosomes to turn those into proteins. And if we have ribosomes, we're making proteins. And enzymes are just a type of protein. And without enzymes, there can't really be any life. So we have to have enzymes. We need a cell membrane, and we need cytoplasm. Everything else is just bells and whistles. This next question, simple cuboidal epithelial cells line the ducts of certain human exocrine glands. It's a lot of crazy words, but just break it down to say there's a type of tissue here, but that's not too important. I need to know that these cells line the ducts of certain human glands. Various materials are transported into or out of the cells by diffusion. We're going to get into diffusion later, but basically you just have to understand we're transporting things into or out of the cells. The formula for the surface area of a cube is 6s squared. Sorry for that typo there. And the formula for the volume of a cube is s cubed, where s is the length of a side of a cube. Which of the following would be most efficient in removing waste? You guys should have said A for that one, because remember that cells prefer being smaller where they can maximize their surface area and minimize their volume. That way they can exchange nutrients and waste faster and be able to get those nutrients to all of the volume of their cell. Number 3, membrane-bound organelles have been an important component in the evolution of complex multicellular organisms. Which of the following best summarizes an advantage of eukaryotic cells having internal membranes? Is it A, eukaryotic cells are able to reproduce faster because of the presence of organelles? B, some organelles such as mitochondria and chloroplasts are similar to prokaryotic cells in structure? C, organelles isolate specific reactions, increasing metabolic efficiency? Or D, compartmentalization leads to a higher mutation rate in DNA, which leads to more new species? The answer for that one is C. While B is true, that organelles like mitochondria and chloroplasts are very similar to prokaryotes, that doesn't answer the question. How is that an advantage of eukaryotic cells? Well, it's not. So we can easily eliminate B. Now, eukaryotic cells actually reproduce slower than prokaryotic cells. Bacteria, they can double each other, some species in as little as 8 minutes. Our cells take hours, sometimes even days to do that. And then for D, this doesn't really have anything to do with DNA. The fact that we compartmentalize, yeah, it protects the DNA, but it doesn't have to deal with mutations. That doesn't quite make sense. We haven't learned about mutations, so we could probably cross that one off. And the last one here, this is going to require a little bit of logical thinking. Researchers propose a model to explain variation in phytoplankton cell sizes in a marine environment. They base their model on the idea that smaller cells absorb nutrients more efficiently. The researchers predict that the mean diameter of phytoplankton cells will change by 50 micrometers for every 5-kilometer increase in distance from the shore because of a gradual decrease in nutrient availability. To test their model, the researchers determined that the phytoplankton cells found closest to shore have a mean diameter of 900 micrometers. Based on the model, what will be the mean diameter of the phytoplankton cells that are found 25 kilometers from the shore? Is it A, 650, B, 875, C, 925, or D, 1150? You should have put down A as the answer for this one. So what they're looking at is they went and found these phytoplankton cells, and when they were the closest to shore, they had a diameter of about 900 micrometers. Their hypothesis is that the diameter is going to change 5 micrometers every 5-kilometer increase from the shore. So we are now 25 kilometers from the shore. What do we predict that size to be? Well, we just have to know that every 5 kilometers, we're going to decrease the diameter by 50 micrometers. If this is based on nutrient availability, and there's going to be a gradual decrease in nutrient availability, our cells are going to have to get smaller. If they have fewer nutrients, they are going to necessarily need a smaller cell to feed. If you have a big cell and not as many nutrients, you're going to get a lot of issues with competition, and if you don't outcompete all the other cells, you're going to starve. So we're going to decrease the diameter 50 micrometers every 5-kilometer increase. So 25 kilometers, that is 5 kilometers 5 times. So we need to just say 5, excuse me for this math problem here. We just need to say 5 times 50. That will give us 250. Subtract that from 900, and you get the answer of 650. So let's get into the actual lecture itself now. These are going to be your objectives. Basically, we're going to go through all of the organelles. You're going to have to be able to tell me what they do, tell me how do they contribute to the overall function of the cell, describe how the structural features of a cell allow organisms to use energy and capture energy, describe the membrane-bound structures of the eukaryotic cell, the parts that make them different from prokaryotes. And then we're going to look a little bit more into compartmentalization and then get into the relationship between the endosymbiotic organelles and their ancestors when they were free living. So we're going to focus our lecture around this idea presented on this slide. The idea that cells have 3 main jobs that they have to fulfill throughout their lifespan. They have to be able to make proteins, make energy, and make more cells. Every single organelle inside the cell is going to fulfill one of these 3 jobs, because without these 3, the cell can't function. If I can't make proteins, I can't make structure, I can't make enzymes, and then my cell is essentially worthless. Cells need energy. We need energy for reactions to occur. We need energy to move things around in the cell. Essentially, without energy, all life is worthless, or it can't occur. And then we need to be able to make more cells. The biological imperative of all living things is that they're able to reproduce. It's one of the characteristics of living things. So a cell has to be able to grow and reproduce to increase its population, or if it's part of a larger organism, to be able to replace dead or damaged cells, or to grow the whole organism, excuse me. So we're going to first look at all of the structures one by one. We're going to start with the plasma membrane. Now, I'm going to go through this one very minimally because we're going to have a whole lecture devoted just to the plasma membrane itself. Think of it as like the skin of the cell. It's made up of these structures here called phospholipids. They have a phosphate head and lipid tails, 2 lipid tails. So one part of it really likes water, the phosphate head, and the lipid tails are very what we call hydrophobic. They do not like water. And this creates this lipid bilayer structure where the phospholipids orient themselves with their tails facing inward to hide from the water that is outside and inside the cell. This allows us to create a very delineating mark of you are outside of the cell or you are inside of the cell. It separates the cell from its environment. And by doing so, it allows the cell to control what comes in and what leaves. It's also important because it'll recognize signals from other cells and allow other cells to interact with each other. Now, we have the cell membrane, and then we have the cytoplasm that is enclosed inside the cell membrane. The first job we're going to look at now is making proteins. So we're going to look at all the organelles that are important to this particular job. You can essentially think of cells as little protein factories. Everything they do is so that they can make more proteins. This is for enzymes, for structure, for things like hormones or messaging systems, so we can communicate with other cells or even other organisms. And the organelles that we're going to look at are the nucleus, nucleolus, ribosomes, endoplasmic reticulum, and the Golgi apparatus. So we're going to go into this much more in depth in a later unit, but I'm going to give you a brief overview of how we make proteins, of protein synthesis. There are enzymes inside the nucleus, right in this part here, that are going to separate the double helix, pull those two strands of DNA apart. A different enzyme is going to read the code and create something called messenger RNA. Messenger RNA is allowed to leave the nucleus, DNA is not. So that messenger RNA leaves the nucleus and finds a ribosome. When it attaches to that ribosome, it's going to actually bring amino acids from the cytoplasm together into the long polypeptide chain that becomes the proteins. The ribosome itself actually creates the peptide bonds between the amino acids. So like I said, this is a very brief overview. We're going to look at every single part of this in detail in another unit, but just so you have a basic idea of what's happening and how we make that happen in the cell. Now let's dive into all these organelles. The nucleus is where it all starts. Without the DNA, we don't know what the code is to make the protein. We need the blueprints to make the structural components, the proteins. So the nucleus itself actually has a double membrane. It has an outer membrane and an inner membrane. We call it overall the nuclear envelope. Another interesting feature is that it has these holes. We call them nuclear pores. And it's from these holes that the mRNA is going to leave the nucleus and go find a ribosome. Unfortunately, it's also used by some viruses. They can exploit this gap, this break in the skin of the nucleus to get into the nucleus and hijack our DNA. Now, aside from that, the inside is covered in what's called a nuclear lamina. Basically, the inside membrane has all of these proteins and fibers that actually give the nucleus its shape. And inside is a structure called the nucleolus. It looks like a dark smudge inside the nucleus itself. Now, why do we need a nucleus? What does it do? It protects the DNA. It creates a very perfect environment where DNA has no enzymes that are going to degrade it. It can't possibly come into contact with an unstable environment. Everything inside the nucleus is highly controlled. So it protects the DNA, which is our instructions for doing anything else in the cell. It's highly important that we keep it separate. And why do we want holes in the nuclear envelope if we need to protect the DNA so much? Like I said, we need to be able to get the mRNA, the copy of the DNA, out of the nucleus so it can go find a ribosome. Now, inside the nucleus is 6 feet of DNA. That means each one of your cells has 6 feet of DNA coiled up and wrapped around inside of it. And it all fits there because it forms chromosomes, these coiled DNA and protein structures that condense this big, massive macromolecule into a smaller version of itself. So the proteins that it wraps around are called histones. And when the DNA wraps around them and forms a complex or a mixture of DNA and protein, we call it chromatin. The full coiled structure is called the chromosome. We're going to go into that a little bit more in depth when we get into our DNA unit. So here's just some pictures. You've got a real electron microscope image of the nucleus itself and the nuclear pores. This is what they look like in a drawing version. And this is the inside of that nucleus, the nuclear lamina, where you can actually see the protein meshwork on the inside that keeps its shape and keeps it protected. So in normal day-to-day life of the cell, you'll be able to see a structure called the nucleolus. It's a really dark, dense kind of like smudge inside the nucleus itself, and it's made mostly of protein fibers. Now this goes away when the cell is dividing, but in the meantime, when it's doing its normal day-to-day cell life, you can see it. What does it do? Why do we have this nucleus inside of a nucleus, essentially? Well, it makes a very specific type of RNA called ribosomal RNA, rRNA. Basically, it makes the nucleic acid portion of the ribosome. Remember that ribosomes were just a nucleic acid and a protein mixture. So without this portion of the ribosome, they wouldn't be able to work and make proteins for us. So essentially, the nucleolus is required for ribosome production. Now, we're still learning more about the nucleolus. It may regulate the cell cycle, which is how cells grow and divide, but that's still cutting edge research that we haven't quite gotten enough research to back up with yet, or enough data to back up the research with. So we've talked about the cell membrane. Last lecture, we talked about the cytoplasm. We've done our DNA, our nucleus, our nucleolus. Now we're going to move further out, away from the nucleus. The next structure that is important for job number one, making proteins, are the ribosomes. First, you need the DNA and the structure that houses the DNA, but next you need the actual things that can read the DNA's code and turn it into proteins. So the ribosomes are a protein and a nucleic acid mixed together, the protein and rRNA. This protein is in a quaternary structure. It's two different proteins working together. It has a large subunit that we call 60S and a small subunit that's called 40S, and that's based on their weights. It refers to the centrifuge speed where you can collect them. Remember, we talked last lecture about self-fractionation and using centrifugal force to pull all of those heavier materials down to the bottom of a test tube. Now, normally they're found free floating in the cytoplasm or cytosol, and they're found attached to the rough endoplasmic reticulum. Like I said before, their job is to make proteins from that DNA code. And they'll do this by doing a dehydration synthesis reaction. Remember, when we remove water from a reaction, we join two different molecules together, two different subunits together. So we'll take one amino acid and another amino acid, and we want to form a peptide bond between them. Well, that's what the ribosome does. It forms that peptide bond, that covalent peptide bond, and we lose water as a result. So, moving on, we now have the endoplasmic reticulum. There's two types, a rough ER that's covered in ribosomes and a smooth ER that does not have ribosomes. So the rough endoplasmic reticulum, the rough ER, is just a bunch of folded membranes. It looks like a membrane maze, this part right here, the darker part. and it's actually part of the outer membrane of the nucleus. So the outer membrane kind of folds away from the nucleus and becomes this network of um, membrane tunnels. The rough ER has ribosomes attached to the surface as well. So wherever there's ribosomes, we're making proteins. So the function of the rough ER is to finalize those proteins. The ribosomes will make them, and the rough ER packages them and helps them fold properly and can even help facilitate forming quaternary structures where two different polypeptide chains come together. They'll often add carbohydrates to the proteins, making them something called glycoproteins. And the interesting thing, whenever you add a carbohydrate to something in the cell, usually you're adding a name tag to it. That's what the little carbohydrate does. It acts as a tag, either to let you in somewhere or let you out. It's a name tag that kind of lets you in with the bouncer of the membrane. So here's an image of how this might happen on the rough ER. You've got a ribosome attached, and it will start to read the rRNA, or the mRNA, and create that polypeptide chain here inside the rough ER. So this is the membrane of the rough ER. And then once it's inside the membrane of the rough ER, it can have little attachments like those carbohydrates. Now, the smooth endoplasmic reticulum is part of the rough ER. It's an extension of the rough ER. So we're still dealing with a continuation of the membrane from the nucleus. Now, this one has no ribosomes on its surface, though. That's why it's called smooth. Its job is to make phospholipids for the cell membrane. It also creates other lipids like hormones. It detoxifies the cell, so whenever toxins come in, like alcohol, it will pass through the smooth ER, and the smooth ER will be able to eliminate those toxins or neutralize them so they don't affect the cell as much anymore. It turns glycogen into glucose in the liver in case we need a quick jolt of sugar in our blood to be able to get through a difficult period like a fight or flight situation. And it's also specialized inside muscle cells to store calcium. So calcium is really important when our muscles contract, and specialized cells like muscle cells have been able to change up their smooth ER to store more calcium so we can do our contractions without having to worry where we're going to get our calcium from. Now, inside the smooth ER, or the smooth ER itself, excuse me, has a really cool phenomenon called proliferation. This has to deal with its ability to detoxify. Basically, since it can make lipids and it's needed to detoxify the cell, if you put a bunch of toxins, like alcohol if you're an alcoholic or a drug addict, into your cells, they're going to actually grow their own smooth ER so they can actually detoxify the body faster, or detoxify the cell faster. This means that if you start to take alcohol or drugs heavily, this is drugs not just recreational, but it could be medicinal, your cells will start to grow their smooth ER, so they'll detoxify that drug faster, so you need more of that drug or more of the alcohol in order to hit the same equivalent reaction. You'll need to feel that same relief from the pain, or to get the same high, you'll need to take more of the drug, but that will cause your cell to increase and make more smooth ER, so it'll detoxify your body even faster, so you need even more of the alcohol. So it makes your buzz, quote unquote, to last a shorter amount of time, so you need more to feel the effects. This is kind of bad when you think about medicinal drugs, because now you're going to have to take more of it in order to have an effect in your body. So now the Golgi apparatus. This is the final one that we'll talk about when we deal with protein functioning and the main job of the cell of creating proteins. Now, the Golgi looks like flattened membrane sacs in a group. I think it looks like a stack of pancakes that are just really fat on the ends. It's like somebody squished them down in the middle. And usually the Golgi is next to the ER because they share a similar function. What will happen is the rough ER will kind of finalize the proteins that are made by the ribosomes, and it will send little packages with these proteins to the Golgi apparatus. The Golgi apparatus sorts these proteins into specialized groups, packages them appropriately, and sends them where they need to go, either somewhere in the cell or it sends them to the cell membrane where they will exit the cell completely. And the proteins are sent out in what we call transport vesicles. So the Golgi, these fat areas on the end, are fat like that because they're starting to bud off. So the ending will start to swell up, just like you're blowing a bubble, and eventually it will get so big that it breaks off from the rest of the membrane and forms its own little transport vesicle, and the proteins will be inside. And that vesicle will then head out to the cell membrane or to another organelle. There's a receiving side. Think of it like a post office. Mail comes in, it gets sorted, and then it gets sent out. The receiving side is called the cis face, meaning it's on the same side as where the material is coming from. The shipping side, or the side where it's going to, is the trans face, and trans means across. Cis means on the same side. So the Golgi will transport these materials. It'll also make carbohydrates, especially if we talk about plants. It's used to help make their cell wall. Now these vesicles, like I said, they bud off from the ER and they arrive on the cis face. And since they're all made of membrane, the vesicles, the ER, the Golgi, it's all made of the same membrane, those phospholipids, those vesicles will fuse with the Golgi pancakes. Then those proteins will get packaged properly. They will move to the end, or they'll move down to the end, and then that will bud off to create new vesicles on the trans face that then leave the cell. So the protein assembly line starts in the nucleus with the DNA. That's the blueprint. The DNA needs to find a ribosome either on the rough ER or in the cytoplasm itself. Then it needs to make the protein, go to the Golgi apparatus to be packaged properly. Then the Golgi creates these little transport vesicles, and that allows the protein to either leave the cell or go where it needs to go in the end. So here's just another picture. You can see the rough ER in blue. It creates the proteins, buds off into these little vesicles that arrive at the cis face of the Golgi apparatus. It unloads all of the materials into the Golgi. They get further packaged, further sorted, and when they get to the ends bud off into new vesicles, and those vesicles can merge with the cell membrane and cause all the contents to leave. And we'll learn how the cell does that as well. So we've covered the cell membrane, the cytoplasm, the nucleus and ribosomes, the ER, and now the G. Now, remember we talked about the endomembrane system and endosymbiosis? This is how we get this whole system of the nucleus, the ER, the Golgi. It all began because a cell folded in on itself. Remember, every cell has a cell membrane. Well, sometimes that cell membrane will fold in on itself and create internalized structures inside the cell, and those ended up becoming the nucleus and the ER and the Golgi, and this is all part of the endosymbiosis theory. So that summarizes job one. We have the endosymbiosis theory created these infoldings of membrane that became our nucleus and our ER and the Golgi. And the whole point of all of those structures is to make proteins. Now, job two is making energy. All cells need energy. All life needs energy. We need to be able to obtain it, we need to be able to store it, and we need to be able to use it. So the organelles that are going to be involved in this part are the plasma membrane, those vacuoles and vesicles I just talked about, lysosomes and peroxisomes, the mitochondria, and of course the chloroplasts as well. So we talked a little bit about vesicles when we discussed the Golgi body. So vesicles, all they are just these tiny little sacs of membrane, the same membrane that makes up the cell membrane, the same kind of membrane that makes up the nucleus, the ER, the Golgi. We all use this phospholipid bilayer. Now, depending on the size and the function, determines whether it's a vesicle or a vacuole. Vacuoles tend to be in plants. They tend to be very large and they'll store water. Vesicles tend to be used to move things around the cell. They'll be much smaller, they'll do their quick job, and then they either become a part of the Golgi or a part of the cell membrane or some other membrane organelle. Their job can be to hold food particles or waste. It can be to transport proteins. It can be to hold enzymes that are going to break down materials. It just depends. Now, with these vesicles, since they're made of the same material as the cell membrane or the nucleus or the Golgi or the ER, that means they can all fuse together. So vesicles fuse to form larger vesicles, they can fuse to the cell membrane itself, and the process of forming new vesicles is called budding. You can see it in this image here. So the cell membrane kind of falls in on itself, pushes in like you're blowing a bubble, material gets taken up in there, and then the membrane reforms behind the bubble. The same thing happens in reverse when it's going to fuse. So they join together like in this image here, and since they're all membrane, they just become part of each other. We're going to go over this in a lot more detail in a couple lectures, so don't worry too much if this is a little above your head right now. We're going to go much more into detail. So the main type of vacuole that you probably learned in 9th grade biology is the central vacuole. And it's found in mature plant cells. Usually it stores water and other materials like that. Really, when you cut open a plant and you see the sap, that's just an aggregate of all of the vacuoles that you've burst when you did that damage to the cell. So the cell can release the liquid stored inside there, and we see it as sap. And it's formed when many different small vesicles fuse together to form one larger one. So what's the job? Why do we have vacuoles in plant cells? Well, they store proteins, they store ions, they store other chemicals that might be necessary for chemical reactions within the cell. Sometimes it'll store waste products or pigments to turn flowers different colors. And sometimes it can even store toxic chemicals. So that way when an herbivore bites into the plant and breaks these vesicles by damaging the cells, it gets this flood of toxic material, and the animal either gets sick or it tastes gross or they even die, and it discourages more herbivores from eating the plant in the future. There's also within animal cells food vacuoles. So what happens is a food item will want to be taken up by the cell. The outer membrane will pinch in to form a vacuole filled with food from the outside. Now, normally we call this phagocytosis, cell eating. There's some cells where this is the only way they can obtain nutrients out in the pond or something like that. But we have specialized cells within our body whose main job it is to go around phagocytizing or eating other cells. We call these our white blood cells. They go around finding bacteria, dead or damaged cells of our own, or even viruses, and they will actually eat them, take them in, and destroy them. Usually what will happen then is that this food vacuole will fuse with another vacuole or vesicle, excuse me, that has some type of digestive enzyme. So now I have this kind of like mini stomach where I have digestive enzymes and low pH and food, and I can break it all down. There's also contractile vacuoles. These are found usually in single cells living in a water environment. They fill and empty with water based on the needs of the cell. So sometimes the cell needs to be able to take in more water because it has a lot of ions. Maybe it's in a more salty environment than it's expecting, so it's going to take in more water, or it's going to get rid of water depending on its needs. You can actually see one down here that's actually emptying of water. There's another one over here that empties of water. So we've talked about the membrane and the cytoplasm. They're kind of part of where this all happens. And we just covered vacuoles and vesicles, and now we're going to move on to the next part. Lysosomes. These are some of my favorite structures inside the cells. They're like the little stomach of the cell. I kind of hinted at them in a couple slides ago. They are little tiny vesicles filled with digestive enzymes. Usually the pH will also be a little bit lower inside this vesicle as well. And it's found only in animal cells. Its function is to digest food. So we will take in food particles inside a food vacuole. It will fuse with a little lysosome and become a stomach with food, essentially. So you have an empty stomach in the lysosome. We fill it with food by the two vesicles fusing, and then the digestive enzymes and acidic conditions of the lysosome will digest the food. We can even use it to digest broken down organelles. So as our organelles grow older or stop functioning or get damaged, we can eat them and recycle the parts. Now, it's made by the endoplasmic reticulum, by the rough ER. So at the end of the rough ER, it'll bud out a little tiny transport vesicle that fuses with the Golgi, gets processed there, then it leaves the Golgi fully ready to work. It then has to fuse with another either food vacuole or some other type of vesicle in order to break down whatever is inside of that. Now, this is a great example of why compartmentalization is important. If I want to be able to digest food, if I don't have membrane-bound organelles like the lysosomes, I'd have to release those digestive enzymes into my cytoplasm. I'd have to make an acidic condition inside my cytoplasm. And if that is in the same environment as my DNA, then my DNA is also going to be degraded. So by creating little tiny compartments within the cell, I can lower the pH within one compartment without disturbing another compartment. So this is where you really see this step up in evolution, where these cells can become more specialized and have a greater range because they can have more functions. You can actually see inside a white blood cells, remember, they eat a lot of other materials like bacteria or viruses inside the cell. So it makes sense that a white blood cell will have a lot of lysosomes because it needs to be digesting a lot of material. Inside here, you can see a lysosome in action. You have the lysosome here, and it's going to be fusing with a mitochondrion. So here's just an example GIF where it shows a food vacuole entering the cell and fusing with a lysosome. So they work best at a pH of 5. This is highly toxic to the rest of the cell. It will create its own pH. It will actually lower the pH inside of that lysosome by taking hydrogen ions from the cytoplasm and pumping them into the lysosome itself. There's actually proteins in the membrane of the lysosome that help this happen. And the proteins inside the lysosomes are activated by this lowering of the pH. They don't take on the proper shape until there's more hydrogen ions inside the lysosome to activate them and turn them on. So why evolve digestive enzymes that function at a pH that's different from the cytosol? Well, I can accidentally have some of these enzymes in my cytoplasm and they won't work because my cytoplasm is a pH of 7. They will only work under the perfect conditions of being in the lysosome. So that way, if anything does happen and these particular enzymes find their way out of a lysosome, they'll be completely inactivated. Kind of cool. And what adaptation does the cell have that allows this difference in pH to exist? That's the compartmentalization that I talked about. So here's some cool GIFs and videos of cells performing phagocytosis and eating other materials, like the white blood cell. You can see all of these little dark structures are bacteria, and this big guy here is a white blood cell that's eating them all up. Same thing here. This is a white blood cell chasing little bacteria around. Now, there's a disease linked with this, lysosomal storage disease. It's usually a fatal disease, which is kind of crazy to think about, that a tiny little lysosome in your cells can cause a whole organism to malfunction and die. What happens is an enzyme inside the lysosome is faulty. There's a mutation in your DNA, and your lysosome enzymes aren't made properly. The lysosome will be able to take in material, but nothing will be digested because the enzymes inside the lysosome aren't folding properly because they've got a wrong amino acid somewhere. So what happens is the lysosome just gets fuller and fuller and fuller, and more material comes in and it won't digest it. Eventually, you're going to have so much waste products inside the cell, or the cell takes on so much that it kills the cell. The cell's not able to get nutrients. You can kind of think of it as like a tumor inside the cell. It gets full of material and it keeps growing and growing until it takes over the whole cell and the cell can no longer function. Another example is Tay-Sachs disease. This is a horrible disease where lipids or fats build up in your brain, and it usually kills the person by age 2 or 3. It's very common in people who are Jewish of Eastern European descent, like my stepfather. So when my stepfather wanted to have kids with my mom, he had to have his blood tested, and so did my mom, to see if they were carriers of this genetic trait, because if they were, they would have had to stop and think, well, do we want to have kids who may end up with this disease? Basically, the lysosomes fill with fats, lipids, that can't be broken down. So the brain gets built up and built up with all these fats. The brain no longer can function because it's being crowded out by fats. And eventually, the baby will start to regress. It will start to develop normally. Then as those lipids build up, the baby will seem to go backwards in time with its abilities, and eventually the baby will die. Another example of a lysosomal storage disorder is actually osteoporosis. Now, our bones are constantly being made and unmade to get at the calcium that we store inside them. Now, when we have these lysosomes that are working way too much, so rather than too little, we're looking at the other side now. They're overactive. They're going to keep breaking down bone even when they're not supposed to. So your bone becomes very brittle and full of holes, and it's more easily broken. Another thing that lysosomes are used for is in something called apoptosis. Fun word, but it unfortunately means cell suicide. Cells will kill themselves or destroy themselves if they have to. So when a cell isn't developing properly, when it's been damaged, there will be triggers inside the cell that cause it to destroy itself so it doesn't accidentally divide and keep perpetuating those problems, passing on those problems to the next generation. A really cool example of when this is a good thing is in normal embryonic development. Sometimes you've heard of people who are born with webbed toes or webbed fingers. Well, that's because the lysosomes didn't do their job to kill cells in between the fingers or in between the toes. So normally the lysosomes will eat away at those cells. They'll kill those cells in between your fingers so your fingers split apart. Same thing with a tadpole's tail. Apoptosis is happening because the lysosomes are breaking open, spilling all of those digestive enzymes out, and killing the cell. Now, if a cell is supposed to do apoptosis, but something stops it, then you often have a situation of cancer going on. Apoptosis is a way for a cell to prevent cancer from taking over the organism. So, like I said, example is with the babies, the little embryos, that webbing in between their fingers goes away because of those lysosomes causing apoptosis. So, peroxisomes on the face are pretty similar to lysosomes. They're membrane sacs filled with enzymes, but they have a different function. They'll break down fatty acids into simple sugars, ones that are more easy for our body to use. It helps detoxify the cell, and it produces peroxide, which I'll talk about on the next slide a little bit. But these structures are found in both plants and animals. Now, we need oxygen in order to produce energy. We'll learn that with our mitochondria. A lot of cells need oxygen to live because we get energy using oxygen. However, oxygen is something that is highly toxic to our cells. Oxygen produces what are called free radicals. These are chemicals that are highly reactive in the cell and can even damage DNA and cause mutations. Peroxisomes can neutralize these damaging oxygen chemicals, these free radicals. What they do is they combine these free radicals with hydrogen ions to neutralize them into hydrogen peroxide. And you think, well, how does hydrogen peroxide neutralize it? Because that is pretty bad for cells, right? Don't we put it on cuts to kill bacteria? Yes, we do. However, peroxide is not very stable. That's why we keep it in a dark brown container and we often have to get rid of it. Because after a few months, especially if it's exposed to sunlight, hydrogen peroxide breaks down into water. So it's a way of taking something very dangerous that can mutate our DNA and turning it back into water and hydrogen. So think about this. Where in the body do you think we have a high amount, a high concentration of lysosomes? Well, white blood cells, as I said earlier, are a great example. Anything where we're breaking down a lot of material. Where might we have a high amount of peroxisomes? Maybe in our liver cells where we're taking in a lot of chemicals that we need help to detoxify all these chemicals that we eat on a daily basis or drink. Now, with energy, when we talk about making energy for the cell, how can we not talk about the mitochondria, the powerhouse of the cell? This is found in both animal and plant cells. It has a double membrane structure because of the theory of endosymbiosis. They were their own cells floating around once, and then they got swallowed by another cell, so they have their own cell membrane, plus the membrane of the cell that swallowed it. It got taken up in a food vacuole and just never digested, and it learned to live in a symbiotic relationship with our cells. Now, the inside membrane has become highly folded, and that is to increase the surface area. More surface area means there's more places for us to do reactions to get energy. Inside of the membrane, it has its own type of cytoplasm that we call the matrix. And another cool thing about the mitochondria is they have their own DNA and their own ribosomes. The ribosomes between eukaryotes and prokaryotes are slightly different. And the mitochondria have the ribosomes that match a prokaryote rather than a eukaryote. So that's even more proof for the theory of endosymbiosis. Now think about, why might animal and plant cells have mitochondria? Well, both animal and plant cells need energy. We need to make ATP. The only difference between animal and plant cells when we talk about energy is where we get that energy from. Animal cells have to consume it from an outside source, whereas plants can make it through photosynthesis. We still have to then take this food and break it down and turn it into a form of energy that the cell can use. So that's why all eukaryotic cells have mitochondria. Their job, like I said, is to make ATP, make energy, through a process called cellular respiration. They convert simple sugars into energy using oxygen. And usually carbon dioxide is a waste product. So that means plants, while they use CO2 to make their sugars in photosynthesis, they also produce CO2 through this process. So it's turning glucose into energy, ATP. We can technically create ATP from any type of food we eat, carbs, lipids, proteins, but chemically speaking, we tend to use glucose because the equation is the best. Now, when we take larger molecules like our food and break them down into smaller ones, and then we use them in this process of cellular respiration, that whole process is called catabolism. Breaking down material to get the energy out of it is catabolism. When we build up molecules, like when we build proteins, that's anabolism. Put both of those two things together, catabolism and anabolism, excuse me, and you get your metabolism, basically the sum of all the reactions going on in your body. Now, what cells do you think might contain a lot of mitochondria? Cells that might need a lot of energy. Maybe like your muscle cells. You need to have a lot of energy in your muscle cells because they're constantly moving. Wherever you have more activity, you're going to need more mitochondria. You're going to need more energy. So similar to the mitochondria are chloroplasts, at least in structure. You have a multiple membrane structure here, except with chloroplasts, they have three membranes. The internal membranes form these stack of coin-like structures called thylakoids, and granum, the whole stack is a granum. Then you have an inner membrane and an outer membrane. So this kind of hints that the chloroplast may have been swallowed by one cell, which was then swallowed by another cell, or it was swallowed twice essentially because it has two outer membranes. Now, the internal fluid, that's cytosol-like fluid, we call it the stroma. In the mitochondria, it's the matrix, and the chloroplast, it's the stroma. Another hint that these are probably part of the theory of endosymbiosis. They have their own DNA, they have their own ribosomes, their own proteins, their own enzymes. The mitochondria and the chloroplast even divide on their own inside our cells. They're going through cell division when our cell is just living its normal life. So we can make more chloroplasts and make more mitochondria within one cell. We're going to talk about these more when we get into our energy unit. The function, though, is to make simple sugars using carbon dioxide from the atmosphere, water from the soil, and sunlight energy. It produces oxygen as a waste product, and we use a green pigment called chlorophyll to capture the light energy to power this whole chemical reaction. So, looking at both of these structures together, you have a double membrane for both, at least. The chloroplast has three. Both have their own DNA, ribosomes, and enzymes. Those ribosomes even match prokaryotic ribosomes rather than eukaryotic ribosomes. Both can divide independently of the whole cell, and interestingly enough, both are involved in energy production. They both actually create ATP, and like I said, we'll talk about that more when we get to the energy unit. The last job that we're going to talk about is reproduction. So the final structures of the cell that we need to learn about help the cell reproduce, which is a requirement for all living things. We need to replace to replenish a or reproduce to replenish a population, to replace cells that have been damaged or died within a multicellular organism, to grow, to repair damaged tissue, etc. We need the nucleus, which we've talked about already, the cytoskeleton, and centrioles. So let's do a quick overview of cell division, just so we kind of know what we're talking about. First, the cell will copy its DNA, and it will make a bunch of extra organelles. Then the membrane will break apart, the nuclear membrane will break apart, the chromosomes will condense, they'll become really tightly coiled, and then special protein structures will grab the chromosomes and pull them to opposite sides of the cell, like you can see in this GIF. Finally, this whole cell itself, the cell membrane and the cytosol, they'll split into two to form two new daughter cells. Now, the cytoskeleton is really important to making all of this happen. It's a network of protein fibers that is actually found throughout the whole cell. So when you're first learning about cells, you see the cytoplasm, the cytosol, and it just looks like a gooey fluid. In reality, this fluid is filled with this cytoskeleton. This skeleton is throughout the entire framework of the cell. When you look at this image up at the top here, all of the green, all of the red, those are all parts of the cytoskeleton, and you can see the entire cell is filled up with it. It's not just empty space or liquid inside the cell, it's a whole framework. The whole job of the cytoskeleton is to provide structure, it gives the cell a shape, it supports the cell, especially when we talk about animal cells where we don't have a nice cell wall to do that. It anchors internal structures, so it keeps certain organelles in a specific place inside the cell. It even acts as a superhighway. Vesicles get carried along these long cytoskeleton chains like it's a particular highway pulling it along. And we even have specialized parts of the cytoskeleton that help cells move. So here's the three different types of proteins that make up the cytoskeleton. You have actin filaments, those are really common in our muscle cells. You have microtubules, and you have intermediate filaments. Now, like I said, these cytoskeletal structures can actually help the cell move. Things like the flagella, the long tail-like structure that moves like a propeller, like in this one here. It helps the cell move around. You have cilia, little hairs that cover the entire surface of the cell that move in a wave-like motion. And you even have little tiny proteins that seem to have feet and walk around on the cytoskeleton. They carry this big ball here is a vesicle, or maybe it's a vacuole or something like that, and it gets picked up by these special motor proteins. This one's name is kinesin, and it walks along part of the cytoskeleton, that little tube at the bottom there. There's also specialized cytoskeleton parts called the centrosome and the centrioles, excuse me. So the centrosome is a region near the nucleus where a bunch of these proteins will organize. Centrioles are pairs. There's these two structures. They're always found in pairs, and they're made of cytoskeleton that are crucial to the division of the cell. We don't quite know how they help us, but we do know that every time the cell divides, these structures appear. They seem like they're helping organize the cytoskeleton fibers throughout the rest of the cell, but we're not quite sure yet. Funny enough, though, these structures are not found in plant cells or fungus cells. They have a cell wall, so they divide a little bit differently. These are only found in animal cells. So this is the whole tour of the cell that you've just taken. We've looked at the membrane, the nucleus, the ribosomes, the Golgi, ER, lysosomes, vacuoles, vesicles, mitochondria, and more. I know that was a really long lecture. Thank you for sitting through it all, and if you have any questions, please reach out to me about it, okay? Have a good one.